we've seen how hosts resist the invasion of pathogens using first lines of defence. But what happens if the pathogens are able to evade those lines of defence? For example, if they enter through a cut in the skin, or if they do manage to bypass the mucus in your respiratory system and get down into the alveoli? Well, we need to look at a process called phagocytosis and the phagocytes that are responsible for attacking and destroying pathogens once they've entered our body. Phagocytosis is another non-specific response. It's not targeted against any particular pathogen, and it's the second line of defence in trying to resist invasion by pathogens. If you take a sample of blood and you ultracentrifugate it, so you spin it out, about 55% of it is plasma, straw-coloured liquid, about 45% of it is cells. The most abundant of those will be red blood cells, the erythrocytes. But about 1% are white blood cells, and the ones we're interested in with phagocytosis are a group of cells known as neutrophils, which we find in the blood, and monocytes, which we tend to move out of the blood and locate themselves in tissues. And these are phagocytes. These are cells which will patrol the body and will engulf and digest any invading pathogen. Another group of white blood cells that we'll look at later are called the lymphocytes. These produce antibodies and the human will respond. But we're not interested in them today. It's simply a comparison between the nuclei, if you look, of the phagocytes, which are kind of irregular and lobed, and lymphocytes, which are round. Is one way to tell them apart. So let's look at the process of phagocytosis. So there are 5 steps. First of these is attraction, then recognition of the pathogen and attachment to the phagocyte, engulfment, which is where the phagocyte actually wraps itself around the pathogen through a process known as endocytosis. Once it's done that, it can form a phagosome and isolate the pathogen in a vacuole inside the cell, and then finally using hydrolytic enzymes and free radicals and hydrogen peroxide, it can digest and break up that pathogen so it's no longer harmful. So let's look at these stages in a bit more detail. The first of them is attraction. This is known as chemotaxis. It's where cells are able to follow a particular chemical trail to find a stimulus. In this case, the little pathogen is giving off some chemical signature, and this larger white blood cell is able to chase it, essentially, and find it. And once it's done that, the receptors on the surface of the phagocyte are complementary to the markers, the antigen markers on the surface of the microbe, on the pathogen. And so by complementary fit, lock and key, these bind. And this is one way that we're able to ensure that our immune system only attacks foreign cells and doesn't attack host cells, non-foreign cells. Once we've got the recognition and attachment, then the membrane of the white blood cell, the phagocyte, will actually start to wrap itself around, forming what are called pseudopodia, around the pathogen and engulf it until eventually form a phagosome, which is a big bubble of membrane in which the white blood cell has isolated the pathogen. Also in the white blood cell you have these structures known as lysosomes. These are bubbles of membrane which contain hydrolytic enzymes, but they can also contain things like free radicals and hydrogen peroxide, pretty nasty things which when they fuse with the phagosome and release into that space will start to digest and degrade the pathogen. Eventually all the soluble products, the carbohydrates and the lipids and the proteins that the cell might be able to use are absorbed, but the antigen markers, just here, are then presented back onto the surface. This is important later on when we look at specific responses. So let's have a little video and see how this process works. Let's take it all the way back. So you can see pathogens attracted, it attaches, it then starts to engulf, eventually forming this phagosome, this bubble around. These are the lysosomes, these little blue dots, which will then fuse with the phagosome and fill it with hydrolytic enzymes, free radicals, hydrogen peroxide, and the pathogen is digested, and eventually anything that isn't needed by the cell is digested, is got rid of. we've seen how hosts resist the invasion of pathogens using first lines of defence. But what happens if the pathogens are able